Well, hello, and welcome to Bio 211. This is AMP1. It is a section at Norwalk Community College in Norwalk, Connecticut. And what you're looking at here is our continuation of Chapter 3, and you're looking at the fact that we have made it to active transport, and here is that purple structure embedded in a phospholipid bilayer, and you can see that on the far left of our cartoon here, you have a sodium gradient and a potassium gradient, meaning they want to move down their concentration gradients. And instead, what you want to see is Mother Nature has to expend energy to move these ions. And the first type of energy she's going to expend is going to be primary active transport, where active transport is going to utilize energy from ATP, be transferred here to the protein, which will then push the material out. But the potential energy stored inside the now channel can be used to allow us to bring two potassium ions in and remove that phosphate. And you'll notice essentially we're back at the beginning. So what's actually going on here? Well, first of all, here's our phospholipid membrane, right? We've got all the sea of phospholipid heads on one side and the sea of phospholipid. So you'll see that we actually have these phospholipid heads here, phospholipid heads here, and inserted as an integral membrane protein is this protein that's named sodium-potassium ATPase. And the reason why we introduce sodium-potassium ATPase is because it's responsible for now creating an electrical difference at the membrane of all cells inside the body. Okay, so I think it's kind of cute. It's a little difficult of an idea to understand, but at the end of the day, you'll have multiple pieces of information inside your brain. The first of which is we actually will utilize active transport that requires ATP. The second is we use secondary active transport, which takes care of the potential energy stored in now the integral membrane protein to now move these two ions of potassium in. But more importantly, you now have a charge at the membrane. Okay? And you'll notice that the net charge at the membrane is you are partially excited on the outside, meaning positively charged, and you're partially negatively charged on the inside of the membrane. Okay? So, let's see what happens when we get rid of that cartoon. Let's move down to the next slide. If that's the case, is it the only type of transport we actually have inside our cells? And the answer to that, of course, is no. Most of the transport that occurs inside of our cells travels as a vesicle. And these are small spherical sacs that are budded off from membranes. An A membrane, it could be any membrane inside the cell. Any membrane. So imagine this, you're inside one part of the cell, you need to move to another part, you're put inside a vesicle, and then you're moved to wherever you need to go to. One form is endocytosis, where you take material into a cell, and it's formed at the plasma membrane. And there are three types of these, right? There's receptor-mediated, there's phagocytosis, and there's bulk phase endocytosis, which I call pinocytosis inside class, right? And receptor-mediated means a ligand will bind a receptor and you'll start a process. Phagocytosis is you'll open up the membrane of the cell and it will engulf something, literally changing the shape of the membrane. Okay, remember we'll use cytoskeletal proteins to do that. And then bulk phase endocytosis, where you'll take in just the fluid that surrounds the cell. And inside of that would be, say, amino acids, some more water, some more ions, some more glucose. The opposite to this, of course, is exocytosis, where we'll take a vesicle and it's going to fuse at the plasma membrane to release their contents into the extracellular fluid outside of the cell. Okay? You could argue that exocytosis occurs from, say, a subcellular organelle as well. An organelle a vesicle that moves into the Golgi apparatus. When it finally leaves the Golgi apparatus, it's exocytosing the Golgi apparatus. You'll see what I mean by that in a few minutes. And then finally, we have transcytosis, where it's a combination of endocytosis and exocytosis, where essentially you travel across the entire length of the cell. Your textbook begins by looking at receptor-mediated endocytosis. The things to pay attention to here is that we actually have the low-density lipoprotein binding to the receptor. Sorry about that. it causes clathrin-coated pits to be formed, and that's what's going to allow for the invagination here of the membrane. That doesn't happen. We don't form a vesicle. The vesicle's formed, then we remove the clathrin. So that means clathrin was required for vesicle formation. And then you actually have the uncoated vesicle fusing with an endosome. And what it's doing is partitioning what's going on here. Where are you going to go? Are you going to get destroyed? You're going to be put inside a transport vesicle. You're going to bind with a lysosome here, and you're going to break this stuff down. The important part about lysosomes is that they have within them high concentrations of hydrogen protons, and, so let's draw that in there, hydrogen proton and enzyme. Okay? The E stands for enzyme. And the enzyme you need to understand, of course, is lysozyme. Okay? But that's pretty much how it works. It's not all that fancy. Okay? Cool. So let's get rid of the cartoon. Bam. See if we can fix whatever I did here. Control Z. Nope. We'll figure it out later. But phagocytosis is when you actually engulf something at the cell membrane. That's pretty straightforward. Same thing, it's now inside of a vesicle. It's now going to go to the lysosome. It's now going to be broken down by hydrogen protons and lysozyme. There are other enzymes in there. The one we teach you about is lysozyme. And you actually have now residual bodies that have to leave the actual cell itself. And here's pinocytosis. Okay, small fluids are brought in. Break it down. You use whatever you need to use and throw everything else out. Okay. Now, the cytoplasm, remember at the beginning I told you there are two components. There's the intracellular fluid that surrounds the organelles. This is where many chemical reactions inside of your cells occur. Okay? The energy is usually released by these reactions, and it's going to allow for cell maintenance, structure changes, function, as well as growth of the cell. Organelles, these are specialized structures within inside the cell. The cytoskeleton is going to be a network, literally of protein filaments throughout the cytosol, and it's going to change the shape, size, location, motility of the cell itself. And we do this by creating microfilaments, intermediate filaments, or microtubules. And you'll see those right here. This is the same cartoon you have inside your textbook. Okay, and for those of you who have a different textbook and you're just listening for the fun of it, this is the polymer of actin that makes up a microfilament. And for the purposes of our textbook, we use keratin as the intermediate filament. There are many intermediate filament proteins. Okay? Keratin makes up this structure that starts at a unique location and then moves inward. Okay? So the polymer is the intermediate filament. The protein is keratin. And tubulin is the protein that makes up microtubules, and you'll see it has a nucleation center, and that nucleation center is this thing that's called the centrosome. Okay? How all of these things work, well beyond what we want to study inside this class, but it's fascinating. If you think about the fact that we want to move the outside periphery of the cell, we would probably engage the actin cytoskeleton first. If we wanted an organelle to move from one location to another location inside of the cell, we might use intermediate filaments, because you can actually see how it's anchored to the side of the cell here and moves inward. Maybe we need the microtubules to move, and you can actually change their length by adding to or subtracting from this. So these can move all the way around. And this makes perfect sense if you've ever wondered, say, how did those chromosomes ever end up at a metaphase plate? Okay. So the centrosome itself is near the nucleus. It consists of two centrioles and a pericentriole material. Okay, we can use them to make cilia, which is move fluids along a cell surface unidirectionally. I should put uni in here. U-N-I. Okay. And flagella, okay, is longer than a cilia, moves the entire cell. Okay, and the example, of course, is a sperm's tail, and that is non-directional. Okay, sinusoidal. But, wherever the sperm is headed, that's the direction it's headed in. And if there's any production flaws in the flagella, meaning you have too many microtubules on one side, it's going to swim around in a circle, and it's not going to be too functional. Okay. Other organelles include the ribosomes, which is the site of protein synthesis, which is what everybody says, right? But what are they? Okay. The rough endoplasmic reticulum, okay, it's a network of membranes, flattened sacs or tubules. You have the rough ER and the smooth ER, and the rough ER itself is connected to the nuclear envelope. And what's going to happen is we're going to use it as the site for the production of various proteins that are not being used inside the cytosol of the cell. Oops, Control-Z. Smooth ER is another network of tubes, does not have ribosomes, synthesizes fatty acids, steroids, and detoxifies certain drugs. Okay? So literally all those bad things you put inside your body that are doing bad things to you, but you really like it, as they're being catabolized, they're being sent to the smooth ER of the cells that they're affecting. Okay? I want you to see the cartoon inside your textbook of what a ribosome is, because you need to understand that we have a large subunit, we have a small subunit, and when we make them functional, you'll see that if this is a tertiary structure of a protein and this is a tertiary structure of a protein, here is the quaternary structure of a ribosome that has the small subunit and the large subunit. And notice in the example here, it's two pieces, but now it's functional. Okay. Let's move ahead. Okay. What is that Golgi apparatus? Okay. These are made up of cisterna, and they modify, sort, and package proteins that are going to different destinations. So what do I mean by that? Most of the modifications are silations or glycosylations. There's some adduct that's added to the protein to give more information as to where it should end up. Think of it as a zip code. Okay? And they're going to be transported by various vesicles to those unique locations. You'll see in a minute when we put the whole story together. Also, we can have these lysosomes. I've spoken about them multiple times now. They're formed from the Golgi complex, so that's the new piece of information here. And they have powerful digestive enzymes inside of them, as well as hydrogen protons. I don't know why that always gets left out. Okay. H plus. Okay. Cool. So now inside the back of your mind, you've created this list of organelles, and now we have to start thinking about how they're all functioning together. Okay. So here is the rough endoplasmic reticulum, and you guys correctly said that, look, we're going to synthesize proteins at a ribosome, and it's going to now be put inside a vesicle, and that vesicle fuses with the entry phase cisterna, and this is modified, and now it's now moved into another vesicle, which is then going to transfer it to the exit phase cisterna. And it has the possibility of being either secreted by exocytosis or inserted into the membrane, okay? And as it's being inserted into the membrane, what's going to happen is this occupies that space in the membrane, okay? Now, what's significant here is that if we're thinking about conservation of membrane, it means exocytosis had to have lost a specific volume of membrane, and endocytosis has to take in a certain volume. This last one over here, these would be proteins that weren't made properly. It's like students who don't study properly. You're put inside a transport vesicle that it's going to go to the lysosome. Not that we actually do this to students or anything like that, but that's what would happen. And if it's going to go to the lysosome, you know what's going to happen to it, right? So I want to start introducing the idea that cells make mistakes when they undergo transcription and translation. And if they're not made or folded properly or processed properly, you have to destroy them. Okay? Cool. Yay. What about these peroxisomes? These are smaller than lysosomes. They detoxify several materials, such as alcohol, which is found inside your liver. It literally breaks down the alcohol, but it can be spit out on top of them, on top of, say, cells to burn holes inside of it. These are usually reactive oxygen species. That's what we call them, right? O2, oxygen species, SEPCIES, right? And what we mean by that is, say, a hydrogen peroxide. It can burn through materials, have a high concentration. It can burn through a lot faster. Proteasomes are like mobile sink disposal units. They continually destroy unneeded, damaged, and faulty proteins found inside the cytosol, inside the nucleus. So they're sent there and they're broken down. Okay? Couple more ideas here before we actually get to how the nucleus works, and then we get to gene expression, right? The first, of course, is going to be the mitochondria. Everybody knows it as the powerhouse of the cell, but what you need to understand, of course, is that it's an autonomous organelle. It has its own genome It is utilizing the oxygen that we're breathing in and intermediates of our glycolytic pathway so it can make ATP for us. It's a waste product for the mitochondria. Okay? It's doing other things with it. And we can change its numbers depending on the amount of activity we actually have. So you increase the amount of activity inside your body, you increase the number of mitochondria inside of your cells. So it's responsive. So there has to be some form of communication between the mitochondria and the human cell itself. Okay? The thing that I make abundantly known inside class, of course, is that if we're thinking about the nucleus itself, these are it's the location where we regulate the normal function of the cell by using genes that have the hereditary units inside of it. And as those genes are transcribed and translated, we then change the function of that cell. But how can it actually happen? Well, you have to create a structure that will store the information. This is the chromatin, which means it's actively transcribed and translated. And you have a nucleolus here, and it's making different structures we're going to use, like those ribosomes. It's going to be making tRNA and stuff like that. And to get it out, we have to travel through the nuclear pore. But remember, the rough endoplasmic reticulum is actually continuous with the nuclear envelope. So we can actually get a partitioning of where things are moving to, right? What else do you need to see here? Well, there's your nuclear pore. Polyribosomes are actually attached at the nuclear envelope as well. So we can have transcription and translation in the nucleus, or at the nucleus. How does that sound? Okay, that's a pretty good place to pause, okay? And before we pick up what is transcription and translation, or as your textbook calls it, gene expression, okay?